So already we're getting a little bit of the analogy of taking one sentence or one paragraph and being able to have it have several different meanings. So you can make several different RNAs from one region of DNA off the sense strand. Okay, now that's been known for a while. That still, I think, is very difficult for evolutionists to deal with. But what we know now, this is what I didn't believe, the sense strand has information also. And not just here and there, not just every once in a while, but consistently throughout the whole strand. At the same place that there's information on the sense strand. So begin to think about that. And this is something, just let that process through. What does that mean? We have information on the sense strand that we're using to make RNA, that we're using then to make proteins, that we're using to make RNA, that we're using for regulatory purposes, that we're using to bend and turn the DNA in its three-dimensional nature. But now we have the exact same activity occurring on the adjacent strand. Now let's look at this just a little deeper and what this means. Remember, the DNA is a two-strand where the nucleotides on one strand are based on the nucleotides on the other strand. They're not independent of each other. So if we have a T on one strand, then it's hydrogen bound to an A on the other strand. So if you have in this case with the nucleotided pairs, let's go back a second. What did I, somehow this silly thing, come on. Okay, with the paired nucleotides, if you have a G, that would be bound to a C on the other strand, on the complementary strand. Now a mutation occurs. Because remember, evolution, it's got to have the mutations to change the sequence, to get new information, to get new genetic capability. You've got to have the mutations occur. Evolutionists love to talk about their mutations. Okay, here you have a mutation occur. So in this case, a mutation occurs that now that G becomes a T. But see, it doesn't do it in a vacuum, because now what happens? On the other strand, it becomes an A. So you have mutated on both strands. Now again, this isn't anything new, but what is new is, let's come back and think about just a second what we are dealing with. It's a little bit like where we have the word live, and the information that would contain, and the word evil, the information that would contain. Notice they're reverse of each other. They're the complement of each other, just like on our two complementary strands. The information on one strand would be the complement of the information on the other strand. But what happens when we change a letter, a mutation if you will, we change a letter in one of the words, so we make it love. Now all of a sudden we have to change this one also, and it becomes evol, which if you look in your Webster dictionary is not a word. So even though you may somehow argue that you have maintained or even changed in an evolutionary sense the informational content, the genetic capability of the DNA on one strand by changing the letter, you've got to take into account, but you change the letter in the other strand too, because this no longer is a non-functional piece of DNA. So what happens with this information also happens with this information. And so we may have maintained this as a word, but we lost what we had on the complement strand. So even if a mutation occurs that evolutionists would argue is beneficial, and we'll talk about that in just a second, the whole thing of beneficial, on one strand, chances are what they've done is they've actually created a degenerative mutation on the other strand. So at best, it's a wash. At best. That's the best they can hope for. And yet they're talking about a mechanism that supposedly takes you from a single cellular organism to the human brain. By this? I don't think so. Okay, so what's that mean in relationship to polyconstrained DNA? That means if we have a mutation on the sense strand, okay, and that strand, of course, is being expressed into various different types of RNA, then those RNA are all affected by that mutation. But see, now we also have a mutation on what used to be called the antisense strand, and all the RNAs coming from that are affected as well. And there is no way by any evolutionary mechanism proposed by the wildest psychedelic dream they could ever come up with that they have a mechanism of how this can occur in the human genome and it'd be nothing but destructive. Nothing but destructive. Because there is too much that happens with that one mutation. And maybe this mutation gives you one group of RNA that actually provides some protein that supposedly is beneficial, but what about all the rest of the RNA that's affected by this mutation? And now you have to double that because now you got all the RNA that comes from this strand that's affected as well. And you think about it as far as a casino lottery. What are the odds of being able to create a mutation here that benefits the organism, and the complementary resulting mutation in this strand also benefits the organism? I mean, the wildest, crazy drunk in Las Vegas wouldn't give you odds on that one. It just isn't going to happen. It's not going to happen. That's why I made the scientists telling me this repeat it several times. Because when I finally began to conceive of this, I realized evolution is now dead. It just hasn't realized it's dead. It doesn't understand the dirt over it means it's buried. But it's now dead. There's not an evolutionist in the world that can touch this. They can't touch. They don't even talk about it. I challenge them on that. They don't want to talk about it. They act like I don't even know. No,